Hello friends, welcome to BMH Learning. This video is about Jaffe test for serum creatinine. First of all, what is creatinine? Creatinine is a breakdown product of creatine phosphate from muscle and protein metabolism. Important thing is that serum creatinine is the most commonly used indicator of renal function. The Jaffe reaction or test is a colorimetric method that we use in clinical biochemistry to determine creatinine levels in blood and urine. Principle: creatinine reacts with picric acid in an alkaline solution to form an orange-colored complex. The reaction is generally known as the Jaffe reaction, and the orange-colored product is called Janowski complex. So basically, proteins in serum are precipitated with sodium tungstate and sulfuric acid. And then the protein-free filtrate containing creatinine reacts with picric acid in alkaline medium to form orange-colored creatinine picrate. The color produced from the sample is then measured at wavelength of 540 nanometer using a green filter. The color and the intensity of the sample is then compared with a known amount of standard creatinine under the same conditions. The color intensity of the sample is proportional to the amount of creatinine in the sample. There is something else very important you need to know. The Jaffe reaction is actually known to be non-specific when used to measure creatinine levels in plasma. It's because many compounds, mainly the proteins, can also form the Jaffe reaction, and thus this can result in the overestimation of the creatinine in plasma. So in order to get rid of these interfering compounds, deproteinization is an important step in creatinine determination of plasma. Materials and chemicals required: test tubes, standard creatinine, picric acid reagent, controls, sodium tungstate, patient samples, sulfuric acid, spectrophotometer, distilled water, sodium hydroxide, centrifuge machine. Procedure: first of all, label test tubes for each control and patient sample to be tested. We are naming these tubes for protein-free filtrate. To each control and patient sample tube, add 1 ml sodium tungstate reagent, 1 ml sulfuric acid reagent, and 1 ml of distilled water. Mix the contents of the tube. Add 1 ml of each control or patient serum to the appropriate tube. Next, centrifuge the tubes for 5 minutes at 1500 rpm. Now this is the protein-free filtrate. Next we are gonna add rest of the different reagents. So for that, let's label the test tubes for control, patient samples, standard creatinine, and also blank. For working standard, that is creatinine standard, add 3.5 ml distilled water and 0.5 ml creatinine standard into this tube. Control, add 2 ml distilled water and 2 ml filtrate made for control in previous steps. Patient sample, similar to the control, add 2 ml distilled water and 2 ml filtrate made from patient sample. Blank, add 4 ml distilled water. Now in all these test tubes, that is standard, patient sample, control and blank, add 1 ml each of picric acid and sodium hydroxide. Keep in mind that the final volume in all test tubes should be 6 ml. After this, mix all the contents and allow to stand for 15 minutes at room temperature. After this, transfer the contents of test tube to an appropriate cuvette and read the absorbance at 540 nanometer against the blank solution. This was all. Thanks for watching.